Good morning, friends. Today I am going to deal about the mechanism of ester-bond hydrolysis using acetylcholine esterase as a candidate enzyme. This will be covered under the topic termed as enzymes in organic transformations, coming under module 2 of bioorganic chemistry course. What is an esterase? Esterase is an enzyme which catalyzes an ester bond. Esters are synthesized or esters are formed due to the reaction between alcohol and an acid. So when you hydrolyze an ester, here first alcohol will be released in the first step. In the second step, acid moiety will be released. A special type of esterase is your lipase. So that I'm going to cover in the next lecture. Because lipids or neutral fats play a major role in any in vivo system. They are secondary emergency energy producers, as well as they are also acting as an insulator. So synthesis and hydrolysis of lipids play a major role in cell signaling, physiology, and metabolism. So as a part of ester hydrolysis, I'm going to discuss this particular lipase enzyme in next lecture. What is the general mechanism of esterase? Please recall the mechanism of action of chymotrypsin, a serine endopeptidase or a serine protease, what I have discussed in my early lecture. The same mechanism is also adopted over here. So here it contains the hydrolysis of esters is mediated in two different steps. Step 1 is acylation, step 2 is deacylation. What we have discussed in chymotrypsin, the same steps, the same mechanism is also followed by any esterase. Then what is the difference between chymotrypsin mediated amide bond hydrolysis and an esterase mediated ester bond hydrolysis? The difference is only one. Under amide bond, the type of the base is amino group, whereas in ester bond, the type of the base is alcoholic group. This is the one basic difference between amide based hydrolysis and ester based hydrolysis, or otherwise the mechanism is almost same. So first step in detail here I will discuss. So the same steps you are going to apply for the candidate enzyme, that is your acetylcholine esterase. So here I have projected a diagram over here for easy reference or as a hotcake reference. So two things happens over here. The esterase enzyme will have a catalytic site. The catalytic in that catalytic site, a particular amino acid will act as a base catalyst, and another amino acid will act as a nucleophile. So here this amino acid will act as a nucleophile, this catalytic site amino acid will act as a base. So there will be a base catalyst and there will be a nucleophile catalyst. So usually the nucleophile catalyst will contain hydroxyl group. Even in esterase enzyme, the hydroxyl group will act as a nucleophile. So here you see, the first we will discuss what happens to the nucleophile or how a nucleophile is generated. In the OH group, you compare the electron negativity of oxygen and hydrogen. The electron negativity of oxygen is very high, so it will grab the electron that belongs to hydrogen, making this particular oxygen as a nucleophile and this hydrogen as electrophile. Now this electrophile hydrogen, where an electron it has lost an electron, so it is in need of an electron. You can see the base catalyst which is present in the enzymes catalytic side. This contains lone pair of electron. So this base will interact, this will act as a nucleophile. It will make a nucleophilic attack over this electrophile, and thereby you can see a protonated base will be generated. Right? Now I have created a nucleophile. I have created successfully a nucleophilic oxygen. This nucleophilic oxygen will attack the substrate. The substrate will be an ester, and any ester will have carbonyl carbon and carbonyl oxygen. So the carbonyl carbon in the ester substrate will be the target atom. So first I have to create an electrophile in the substrate. I have to create an electrophile carbon. So how that is possible? You see step 3 over here. Now you compare the electronegativity between oxygen and carbon. The electronegativity of oxygen will be very high again when compared to carbon. So the electron that belongs to carbon will be grabbed or attracted by the oxygen, creating nucleophilic oxygen and electrophilic carbon or carbocation. Now this carbocation is ready for the attack. I have generated already a nucleophilic oxygen in the enzyme's catalytic site. This will make an attack over here. So thereby you see tetrahedral intermediate will be formed. So this is the first phase. Now you see what happens to this particular. Now I want to hydrolyze this because already I have mentioned in the first phase of the reaction alcohol will be released. You can see alcohol is released. In the second phase acid will be released. So the alcohol will should be released over here by hydrolyzing this particular bond, right? Now you see here, right, the electron of this particular carbon, right, again it will be shared over here from oxygen. The electron will be shared over here, right, and the electron will be delocalized over here, so making now this oxygen as nucleophilic in nature, right. Now you can see third amino acid in the catalytic site of your esterase enzyme. So this is one amino acid, this is second amino acid, this is third amino acid. So the third amino acid is a protonated base. What happens, you see, again the electron that belongs to hydrogen will be delocalized to this particular base atom, making this atom as nucleophile. Usually this atom will be either nitrogen or it will be oxygen. Assume in this case this is a nitrogen, right? So successfully I have created a nucleophile over here. Now what happens to this left out electrophile? Now it is ready, it is deficient in electron, it is in want of an electron, it search for an electron. Now you see already here in the substrate I have created a nucleophile like oxygen. It is having a lone pair of electron. It will, this oxygen will make a nucleophilic attack over this electrophilic hydrogen, and thereby an alcohol will be released. So the first step is formation of tetrahedral intermediate and release of alcohol. This is what, now this part we are telling as an acylation, right? Successfully I have created an acyl group, enzyme acyl group. Now in the second step I want to degrade the bond between you can see this bond. So I want to degrade this particular bond. The enzyme acyl should be degraded and the acyl group should be released as an acid. So that is the second phase, what we are telling as a deacylation phase. For this, I need again a potent nucleophile, but here the nucleophile is not coming from enzyme's active site because the enzyme's active site nucleophile is under already bond, right? It is bonded with the substrate, so there is no suitable nucleophile distributed in your enzyme's catalytic site. So system has to search for a potent nucleophile. That potent nucleophile is nothing but a water. So again, please recall the chymotrypsin mediated reaction. So there even there under deacylation mechanism we have used water as a nucleophile. The same mechanism is also adopted for ester hydrolysis. In deacylation process, water will be acting as a nucleophile. So how it is acting? You see the reaction mechanism. You can see the structure of water projected in magenta color HOH. You compare the electronegativity of hydrogen and oxygen. Again, electronegativity of oxygen will be very high. So the electron that belongs to hydrogen will be grabbed by this oxygen, making this oxygen as nucleophile. So that could be the first stage, right? So in the second stage, what happens? You see, I have to create a suitable electrophile in the substrate. The same mechanism, electrophilic carbocation will be generated by delocalizing the electron that belongs to carbon to oxygen, making oxygen as a nucleophile. So nucleophilic oxygen and electrophilic carbocation is generated in the substrate which is attached to the enzyme. What is the third stage? Now the catalyst, that is your water, which contains nucleophilic oxygen, will make an attack. So thereby it will make an attack, and simultaneously the nucleophilic oxygen it will make a nucleophilic attack over the protonated base. Remember, already we have generated a protonated base. Please see the first step, already we have generated a protonated base. So this proton will be attacked by the nucleophilic oxygen. So Hb plus, remember, yes. So you can see Hb plus. So this nucleophilic oxygen will make an attack over here. So as a lot you can understand, right? So R, right, C O, this lot C O O R H, right, R C O O H as a lot will be removed as carboxylate or carboxylic acid, and you see the rest, the enzyme will be regenerated. So this is under deacylation. So what is the specialty of today's enzyme acetylcholinesterase? I will not discuss much more about acetylcholine. This you can, if you are interested, you can study under neurobiology course. There is a neurotransmitter termed as acetylcholine, which is distributed in neuron. Acetylcholine is the temporary message which carries the message from one neuron to another neuron. This first neuron will be termed as pre-neuron. Second neuron will be termed as post-neuron. Any neuron will have a knob that will be termed as pre-synaptic knob. The post neuron will have again another knob that will be termed as postsynaptic knob. So knob is the end, nerve ending. So what happens, you can see there is a junction or space between any two neuron. The space is actually termed as synaptic cleft or synapse or synaptic space. You see the message will be in the form of ions. And what happens when this neuron try to pass the message to this neuron, there is a space. So automatically there is a chance of leakage of message. The ions will be leaked and it may be distributed in the space. So the second neuron may not get the message. So how to solve this problem? So there should be a messenger which should carry the message from neuron 1 to neuron 2. That messenger is nothing but your acetylcholine. So you can see this acetylcholine will interact with the receptor distributed in any synaptic knobs or in any neurons. When acetylcholine interacts with the receptor, this is nothing but a gate, closed gate. When acetylcholine interacts with the receptor, the gate will open and the message in the form of sodium ions will enter into the second neuron, right? So as a result, successfully the message from neuron 1 is transmitted to neuron 2. So please watch this YouTube. You can understand how a synaptic transmission occurs between any two neurons, right? So very carefully you watch this video. This will act as a prelude about neurotransmission. Now what is the problem over here? The problem is, in order to solve the message problem, I have used acetylcholine. But if this acetylcholine is going to interact with the receptor for a long time, the message will continuously trigger, right? When there is a start, there should be always stop. When there is a origination point, there should be always an end point. So what is the end point? Now I have started the message. The message is continuously disseminated. I have to stop the message. How to stop the message? I have to remove this acetylcholine. How to remove the acetylcholine? There comes the help of your enzyme acetylcholine esterase. So acetylcholine esterase will hydrolyze acetylcholine. Now acetylcholine esterase again will be distributed in any neuron. Any neuron surface will be studded or anchored by acetylcholine esterase. So this acetylcholine esterase will catch hold this acetylcholine. It will degrade. You can see the mechanism. So this is the ester bond. So the ester bond of this neurotransmitter acetylcholine will be hydrolyzed by acetylcholine esterase. And already we have discussed in the second phase of the deacylation mechanism, water will be used as a nucleophile. So where this ester bond will be degraded, and acid and alcohol in the form of choline will be liberated. So thereby I am stopping the dissemination of the message. So in that way your acetylcholine esterase play a major role. If there is any lack in the activity of your acetylcholine esterase, acetylcholine will not be removed, so the message will be continuously passed. This leads to many neurodegenerative disorders or neuronal related disorders. So, what is the if you, before understanding the mechanism of action of acetylcholinesterase, we have to understand the structural characteristic features of acetylcholinesterase. The active site of or catalytic site of your acetylcholinesterase is too narrow, and the catalytic site is comprised of two different sites. One is anionic site or anionic binding site, another one is estratic site. What is the speciality of anionic site? It interacts with the quaternary ammonium cation. This interaction is not directly, indirectly this anionic site helps to stabilize the substrate. Please you see the substrate. So this is the substrate. You can see a cationic ammonium ion N plus. So if you want to stabilize this substrate in an enzyme, right, we need a negative charge in the enzyme. So here it is chlorine, you forget about chlorine. So in order to stabilize this N plus, we want a suitable negative charge, right? Such a kind of negative charge amino acids will be lined or it will be placed in the catalytic site of your acetylcholine esterase. So in that way, the anionic amino acids which are distributed in the catalytic site of your acetylcholine esterase interacts with uh, choline of your substrate. Then what is the role of estratic site? It is the site which is responsible to hydrolyze your ester bond. So the mechanism is similar to any kind of serine hydrolase. So the active site gorge is made up of three different amino acids, catalytic triad. Already we have discussed under chymotrypsin. Please recall, chymotrypsin's catalytic triad contains three amino acids. One is serine, another one is histidine, third one is aspartate. The only one difference over here is even acetylcholine esterase contains serine and histidine, whereas instead of aspartate here we are having glutamate, and the location is different. So serine 200, histidine 440, glutamate 327. These are the catalytic triad amino acids which are distributed in acetylcholine esterase and which are majorly responsible for hydrolysis of ester bond of acetylcholine. So apart from catalytic triad amino acids, few other amino acids also play a major role in stabilizing the acetylcholine substrate, and thereby those amino acids in the enzyme help the catalytic triad amino acids to hydrolyze the substrate very effectively. So one such amino acid is aspartate 443, right, where it is responsible to form hydrogen bond with water. Again recall, water is an important nucleophile which play a major role in the second phase of the reaction. Another important catalytic site amino acid is glutamate 199th position, which stabilize the imidazole group of your histidine, which is a part of your catalytic triad. And as I said earlier, the quaternary ammonium ion which is present in your substrate is not directly interacting with the anionic site, but it interact with the aromatic residues, where one such aromatic residue is your tryptophan 84, where this tryptophan 84 straight away interacts with the quaternary ammonium ion, so that your acetylcholine will be stabilized in the enzyme's catalytic site pocket. Again you can see these catalytic site amino acids in acetylcholinesterase: tyrosine 337, phenylalanine 338, tyrosine 133, tryptophan 84. These sort of amino acids, they are highly hydrophobic in nature, and they are responsible to stabilize your substrate acetylcholine, because acetylcholine also contains more number of hydrophobic moieties. Please watch. This is the substrate acetylcholine. It contains, you count how many amino methyl group. So methyl group 1, 2, 3, 4, 4 methyl group, and then here one methylene group CH2, another methylene group CH2. So at least 4 methyl group and 2 methylene groups which are highly responsible to create a kind of hydrophobicity. They are all hydrophobic in nature. So if you want to hold your substrate acetylcholine very effectively for hydrolysis, a system has to generate a hydrophobic interaction, right? So such kind of hydrophobic interactions will be generated by these set of amino acids which are present in the catalytic site of your acetylcholine esterase. So your acetylcholine, your substrate will be fixed in the binding pocket active site for effective hydrolysis. Apart from that, these three amino acids, glycine 118, glycine 119, and alanine 201, play a major role for the formation of oxyanion hole, which is a part of phase 1 acylation of your acetylcholinesterase mediated hydrolysis. Here I have projected the three-dimensional structure of acetylcholinesterase for easy understanding. You can see the red color, yellow color, and gray or white color. These three spheres indicate catalytic triads: serine 200, histidine 440, and glutamate 327. And you can see the blue color sphere ball indicates the tryptophan 84, which is responsible to interact with the quaternary ammonium ion of your acetylcholine. And you can see these three set of amino acids. Violet color, pinkish violet, glycine 118, 119, and alanine 201, which is responsible for the formation of oxyanion hole. And the orange color spheres, aspartate 443, glutamate 199. Already we have discussed aspartate 443 is responsible to stabilize water, which is acting as a nucleophile. Glutamate 119, which is responsible to interact with the imidazole group of your triad histidine. So you can see, even though these set of amino acids were distant, the position says it is 84 tryptophan, whereas aspartate 443, here you can see serine 200. Even though amino acids were distant, because of folding of the protein, these amino acids were brought very close or in proximity with each other, which creates a small narrow cleft space where here your acetylcholine will be fixed. Now that's what it was projected over here. This is the cleft. Please, you see the cleft over here. This is the cleft. Now this is the space where you can fit your acetylcholine. Now your acetylcholine will be fitted over here for effective hydrolysis. So already I have discussed the mechanism of action of ACE. I am not going to interact much more about this. I'll give a prelude. Two steps will be there: acylation and deacylation. The reaction mechanism is almost similar to chymotrypsin or to any other serine hydrolase or serine protease. So here, this is the overall mechanism. So this is the first stage, creation of tetrahedral intermediate on oxyanion hole, where alcohol will be released. So in the second stage, your acid moiety will be released and enzyme will be regenerated. So for a hot cake or for easy understanding, I have projected a handwritten picture over here, image. You can see catalytic triad amino acid of your enzyme acetylcholinesterase, glutamate, histidine, serine, right? So here you can see the purpose of glutamate is to stabilize your histidine through hydrogen bonding. And what happens first? I have to create a nucleophile, right? So enzyme catalytic site amino acid nucleophile should be generated. So serine nucleophile should be generated. So for this, again, the story repeats. The electron of hydrogen will be grabbed by oxygen. So as a result, serine oxygen will be nucleophile. It will act as a nucleophile. This could be step 1. And what happens to the released electrophile proton H plus, that will be accepted by the base, right? Please recall, we have discussed in the first phase presence of base catalyst and presence of nucleophile catalyst is very important. So that we have discussed in the early slide. So histidine will act as a base catalyst and serine will act as a nucleophile. So in the second stage, the released electrophile will be grabbed or attracted by the nitrogen which is present in your histidine. Nitrogen is a potent nucleophile. So in the third step, automatically a tetrahedral intermediate will be formed. For that, I want to create a nucleophile oxygen. So then only an electrophile carbon will be generated, a carbocation. Now this nucleophilic oxygen of serine will attack the carbocation which is present in the substrate, right? So this is the substrate, right? And thereby the potent oxy anionic hole and tetrahedral intermediate will be generated, right? And slowly this tetrahedral intermediate will be collapsed and your alcohol will be generated, right? So in the second step, deacylation, again here I have repeated, is nothing but reverse of your acylation, right? So here you can see I have to create a nucleophile. For that I have to create an electrophile, again a nucleophile, and again an electrophile. So the hydrogen is acting as an electrophile, this oxygen will act as a nucleophile. If carbon is acting as an electrophile, this oxygen will act as a nucleophile. Almost in many biochemical reactions, you can see oxygen and nitrogen of catalytic site amino acids will act as a nucleophile. Your hydrogen and other atoms which are possessing lesser electronegativity than hydrogen or oxygen will act as an electrophiles. So we can see 1 2 3 4 slowly reverse, and you can see here, so here there will be methyl group, this bond will be hydrolyzed, degraded with the help of water. Here I have not, you can see water over here, so this is water nucleophile, and slowly this uh, linkage between enzyme catalytic site or this acyl will be removed. That is what we are terming as deacylation. This removal of acyl will be released in the form of acetate. Now remember, we are speaking the substrate as acetylcholine. Acetylcholine is the substrate. So already choline is generated as alcohol. Left out is acetate, that is also generated over here, and the rest of the catalytic site amino acids will be regenerated as such. Now it is ready for the next cycle. Now second acetylcholine can interact, again acylation, deacylation, right? So friends, if you understand the mechanism of action of sorry, chymotrypsin, the same mechanism of action is also adopted over here. Synopsis, two steps will be there, acylation and deacylation. In the first step, the serine moiety, serine hydroxy group will act as a nucleophile. Histidine imidazole will act as a base. The second deacylation, water will act as a nucleophile, and the tetrahedral structure, which is attached as an acyl form to the enzyme, will be hydrolyzed, and the acid moiety will be liberated. So hope this lecture may be simple, even if it is complicated. If you say that this lecture is complex, that means you are unfamiliar about the fundamentals. So again, please read the fundamentals. Please read the fundamentals. What is a nucleophile? What is an electrophile? What is an ester? What is the difference between an amide bond and a normal ester bond? How amide is formed? How a normal ester is born? Normal ester bond is formed. So by having these kind of simple fundamental fact, if you would have understood the mechanism of action of chymotrypsin, automatically this could be easy for you, right? So uh, thank you, thanks a lot. If there is any query, you can write to me, you can email me, or through live session, I will interact and I will clarify all your doubts. Thank you friends.